You could say 2025 was a big year for March of Dimes data. You could say a special March of Dimes dataset was chosen for a competitive National Science Foundation AI program. You could even say March of Dimes launched its first AI assistant called ChatPTB or ChatPreTermBirth. And you'd be right. All those things are true. And one woman played a key role in making them happen. Her name is Dr. Tomiko Oskotsky, and she's here with me today. This episode is about March of Dimes data's big moment. Dr. Tomiko Oskotsky is an integral part of the March of Dimes Prematurity Research Center at the University of California, San Francisco. Along with the PRC's principal investigator, Dr. Marina Sirota, Dr. Oskotsky co-directs an indispensable part of the center, the March of Dimes database for preterm birth research. It is there in a system of computers, hard drives, Google Drives, clouds, briefcases, and desks that the holy grail of March of Dimes scientific outputs are stored. The data from every paper published by a March of Dimes PRC since 2015. But they aren't just stored there, coveted by March of Dimes scientists and kept from the world. They're uploaded online. They're shared in their entirety. They're open to curious minds, the minds of engineers and mathematicians and doctors and students and AI experts to explore and use for free. All in the name of making big discoveries for the health of moms and babies together. And Dr. Oskotsky helps oversee it all. Tomiko, welcome to the show.
Thank you, Alessia.
You've been on once before, Tomiko. I know we were talking about that with Dr. Golub and Dr. Sirota, talking about the Dream Challenge, and we will talk about that a little later in our recording. But it's great to have you back. Welcome to podcast number 2, Modcast number 2.
Yeah, and it's great to be back. And it's wow to think back how that was really wonderful talking to you about the Dream Challenge, and to think about how much time has passed since then. It's a bit mind blowing.
Absolutely. I think it's been at least a year. Now,
Yeah.
This 2025 and probably a couple months at the end of 2024, it's been a big year because we're recording this in October 2025. It's been a big year for the database that you co-direct and for your team at the PRC, especially because this year, 2025, marks 10 years, 1 decade of data collection for the database. Even though it was technically launched 9 years ago, you guys have been collecting data for 10 years. So congratulations on that anniversary.
Yeah, no, thank you. It's quite a milestone, and it's really amazing for me to look at the trajectory of the database in that amount of time and see how much it's really rapidly grown, especially in the past few years.
Yeah, absolutely. And so I want to give a very quick overview of the database, which is a gold mine. So this is a repository of every piece of molecular data generated by a March of Dimes PRC since 2015. There are 79 studies in there right now, actively growing every day, every week, every month. More than 46,000 experimental samples from nearly 31,000 participants. It is the only public multi-omic resource for preterm birth research in the world. And it brings together multiple types of biological data, including DNA, RNA, immune system markers, and the vaginal microbiome. It underscores March of Dimes' commitment to open science, collaboration, and accelerating the pace of research progress to end preventable preterm birth. So a lot of unique data in the database, obviously, but perhaps the most important part, Tomiko, is that it's being shared publicly with the world, with other experts who may not have had access to it otherwise. This is something Emery, our chief scientific advisor, is very passionate about and feels strongly about. I know you and Marina do as well. So talk to me in your own words. Why is it important to share this data? What's the point of all this?
Yeah, no, absolutely. I believe all of us in the prematurity research centers are passionate about ensuring that the work we are performing, the data that we're generating, are able to be reused by others and, lead to exciting, novel discoveries. And for us, open science, it's this idea of research inputs and outputs, like data and code and methods and papers, that should be accessible as possible, like as widely as possible to many others, so that they can verify and reuse them and build on them. When we share all of this data, we can turn single studies into compounding knowledge, and the results can get checked, new questions get asked, and discoveries move from being interesting to impactful at a much faster pace. And so this is why we all do what we do.
I love that, from interesting to impactful. Yeah, that's beautiful. That's wonderful. Okay, so one of the both interesting and impactful data sets in this repository, maybe the most special data set in the repository, is a unique vaginal microbiome data set. And this is your first big win. And this is a big one. Earlier this year, recently, I think it was earlier in September, the National Science Foundation announced
That's right.
that
Mm-hmm.
this data set from the March of Dimes prematurity research, from the March of Dimes database for preterm birth research, was chosen for inclusion in a competitive AI, artificial intelligence pilot program called the National Artificial Intelligence Research Resource, or NAIRR. This data set was just one of 10 community data sets chosen for the pilot, so it was competitive. And the pilot is a public AI database aimed at advancing the nation's AI workforce. And the March of Dimes data was the only chosen organization representing pregnancy or healthcare. So we were alone in that field. The inclusion of this data, put our data on the national AI stage, dramatically expanding the data's reach beyond preterm birth community to spur more and faster and more frequent scientific breakthroughs. It was a real nod to the caliber of the organization's research, much of which you curate and organize, Tomiko, as part of the database, and its ability to harness AI to help understand, predict, and prevent preterm birth through new diagnostics and therapeutics. And at the PRC, you guys are doing a lot of work using AI, drug repurposing, electronic medical records. So certainly you're one of the strongest AI components to the PRC network. And so in other words, the inclusion of this data set into this pilot was a very, very good thing, and a very good day for all of us at March of Dimes when we found out that we were included. Tell me what it was like for you and Marina when you found out that this database,
Yeah,
which was founded in 2017, well, pretty recently, was
yeah.
included in this very prestigious honor.
Yeah, no, absolutely. And I'll tell you, what was it like? It was incredibly thrilling when we got the good news. And actually, it was a bit more recent. What happened was back in the fall, a colleague, Kate Heitz with Peraton, let us know around the fall. I want to say it was early or late fall that the NSF was looking for individuals and organizations to contribute AI-ready data sets for inclusion in this NAIR pilot.
And that was a
And
year
as
ago, right, Tomiko? 1 year ago?
Yes, yeah.
Yeah.
Oh my goodness, what's so funny as we talk about
Yeah.
it, to me it feels like yesterday, but you're right, it was a year ago. Oh my gosh. Yeah, time really does fly. So it was about a year ago, and we learned about this, and we realized that in this call that we had, as you mentioned, this Dream Challenge data set. This was a couple of years ago already, or gosh, I guess it's been three, where we were able to use this aggregated vaginal microbiome data set for a machine learning challenge. So in a way, it was primed for this call. It was AI ready, but they had this checklist, this tremendous checklist that we had to make sure adhered to the various criteria that they were looking for. So we worked with our Peraton colleagues and actually individuals across UCSF to help with some of the more nuanced aspects to ensure that our data set indeed met these criteria. So after all this rigorous work, we realized it did, and we submitted it for consideration. And that was around, I'd like to say, February. And we got the news shortly before our meeting with Emre Selly and Jonathan Cherry for our annual site visit at UCSF, like literally a week before. So we were just busting at the seams to share this amazing news in person when they were all on site for our visit. So that was tremendously exciting to know that the National Science Foundation recognized the value of our data set. As you noted, it is amongst the 10 data sets that were selected, ours is the only one that really represents women's health. It represents a data set from individuals. And so it's incredibly it's a really amazing recognition. And it also is one by our now being able to feature this data set on the NSF's NAIR platform, we have just tremendously expanded the reach of this data set being able to be leveraged by others to include it in their work to, come across their discoveries. So it's just all the way around incredibly exciting.
Yeah, exactly, Tomiko. The first win was, oh my gosh, our work has been recognized and chosen. The bigger win, arguably, is that our work can now fuel discovery around preterm
Exactly.
birth. So, talk to me a little bit more about that. What potentially could happen here now that this data is accessible to the greater community? What would be a best case
Yeah,
scenario for you?
I think best case scenario would be, I think there have been a lot of single studies that have looked into looking at associations between the vaginal microbiome and preterm birth. I know that preterm birth is definitely multifactorial, but the vaginal microbiome has specifically been seen as something that's involved. It's associated. And these single individual studies previously have identified certain aspects that could be interesting. By our ability to aggregate this into a larger data set, definitely we increase the statistical significance, the power of being able to explore this data. And the machine learning challenge that we had this data set involved with also, we could say that we saw a signal there as well, saying there are things that are contributing here, or associated, I should say, associated. But I think what by now making this data set available more broadly, individuals can explore this even further. They now have greater power to explore this as well. So we've taken it from being a sort of individual or site, one site kind of looking at this problem to a national challenge, to an international challenge where they've applied machine learning techniques to try to explore this. And now we've expanded the reach to far wider than that, where people can look into this, looking into, how is the vaginal microbiome potentially involved in preterm birth and potentially other conditions as well.
Yeah.
So it's really like it's phenomenal to know that it's being used for this purpose by individuals who are interested, as well as one of the NAIR pilots' goals is also in education, in training individuals. And so by
Mm.
using this data set, they're making individuals aware of the issue of preterm birth, that there needs to be more understanding, that there need to be therapeutics. And so it's beyond the data set itself, it's the expanding the call for help to individuals who now recognize that this is an issue that needs attention.
Yeah, absolutely. A quick question for you, Tomiko. Is this kind of a pipe dream? I really hope that other researchers around the world get involved. Let's make it free. Let's make it available to them, and they'll, take the time out of their regular 9 to 5 or 9 to 9. People work long hours to log online and explore something that they're not even researching themselves. Or is this actually happening actively all around the world already, where researchers are hungry for free data that is AI ready and they can tinker with?
I do believe people are hungry for this kind of data. When you look at websites like Kaggle and others, there are lots of people who are getting involved in these AI challenges. And it's not necessarily because of the monetary or other compensation. It's really because people they want to help, they want to do something. And, and machine learning is exciting and AI is exciting. But when we can draw those people who have the skills or the desire to help, if we can draw them into this field, that is amazing too, because then we have different insights, different skill sets, different talents that are coming in here. And that's what we discovered with our Dream Challenge. We had individuals who won and came to present their work at that conference in Las Vegas. That was the prize. And most of those individuals have never worked with vaginal microbiome data. They didn't know uh, about preterm birth that much. Like they knew AI and they were thrilled to be able to apply their skills to this. So I do believe that there's a lot of people out there with skills, with a passion to do good, who by seeing data sets like these available they're going to be able to help us move forward.
Yeah, amazing. Wonderful. Okay, well, for anybody that didn't clue in yet, that's listening or watching, the dream challenge that I mentioned at the beginning of the episode and Tomiko has referenced multiple times is made of this data set. It is comprised, the foundation of this challenge is from looking at the same data set that won the NAIR pilot, that was included in the NAIR pilot. This is the vaginal microbiome data set. So let's talk a little bit about this. So, Tomiko, you've said about this data set, you've said numerous times that this, one of the key reasons it was chosen is it was because it was AI ready. It was a machine learning challenge in terms of the dream challenge. What does it mean to be AI ready? And tell me how the team, how your PRC team managed to get it that way. I know there was a big challenge involved and a super sleuth on the team cracked it. So tell me what
Sure.
AI ready means, what that challenge was, and how you managed to solve it.
Sure. Well, one of the things about AI is you need a large data set. You need something beyond just like a few samples or a few, dozens of values. You need something much larger. So that's one thing about our data set is quite large. As I mentioned, with vaginal microbiome studies, they've previously been kind of one one cohort at one location. It's been technically very challenging to aggregate, to harmonize vaginal microbiome data across sites, across studies. It's just been a technical feat that's been challenging to overcome. The fact that this data is so large and harmonized, it's also richly labeled. There are explicit labels for outcomes, sample timing, key clinical covariates like maternal age and BMI and parity, as well as a lot of the microbiome features that are included, like community state types, phylotypes, taxa. There's a lot of features. It's a feature-rich data set. And it's very well structured and organized. So pretty much it's shovel ready. It's not like people have to do a lot of wrangling, and like that's one of AI scientists' nightmares, is having to like wrangle data. And this data set is pretty much like shovel ready, which just makes it a dream to work with. No pun intended.
Yeah,
And you
absolutely. It's
mentioned,
like it's like buying vegetables at the grocery store that are already pre-cut. You know, one of the most time-consuming and annoying parts of cooking is cutting and chopping and all that, unless you're alone and you have many hours. But yeah, and tell me, and what was that technical challenge, Tomiko, that you mentioned, that it was difficult to harmonize the data from each of these different studies? Why was that?
yeah, so exactly. So harmonizing, for people who may not be as familiar, it talks about bringing multiple different um, pieces of data from different sources and bringing them together into one large data set that's all standardized and can be kind of worked with all together. And for different types of molecular data, like transcriptomics, for example, that's much easier to accomplish. There are actually already methods for doing that. But for microbiome data, that hasn't existed. But we have a genius colleague, Dr. Jonathan Golub. He's an infectious diseases clinician as well as a physician scientist who's incredibly computational savvy. And he developed a way, it's called Maliampi, it's a computational tool that's able to bring vaginal microbiome, oh, sorry, not just vaginal, but microbiome data sets from different body sites and bring them all together into one harmonized data set. So that's what we used, is for this challenge, we used his wonderful Maliampi tool to aggregate and harmonize almost nearly a dozen data sets all together into one large, amazing data set.
Fantastic, yeah, and it's much more complicated than that too, right, Tomiko? I know that you gave us a high line kind of explanation, but when Jonathan explained it to us on the podcast, the Dream Challenge episode, and to me at various times, it's quite complicated. Does it have to do with a protein or a genetic coding thing, or
Right.
what was it again?
Yeah. So
I-19.
this,
What was the number there that we were talking
yeah,
about?
yeah, yeah. No, that's a good
There was a
question. So
number.
yeah, the type of vaginal microbiome data that we aggregated for the Dream Challenge is called 16S. It's a
That's it.
region of yes, it's a region of the of the ribosomal RNA that is able to be, and there's different regions of this 16S that it is used in different studies, and that's what makes the harmonizing challenging. Actually, that's one of the things. So this 16S data the sequence 16x data using the different variable regions is what we used in our challenge, and what Jonathan was able to harmonize regardless of which variable region was sequenced. So that is what enabled our challenge, because without the data, we'd have had nothing.
Yeah, beautiful. Now, the other reason that this data set is special is because, as you mentioned, it is large. So there's a high number of samples. It's also diverse, comes from a diverse population. It's got more than 3,500 vaginal swab samples from over 1,200 pregnant individuals across the PRC network. Is this really, are these types of numbers super rare in this field,
Yeah,
Tomiko?
yeah. Oh, yeah, yeah. Up until our challenge and when we brought this data set together, the kinds of numbers we were seeing were a fraction of this. And they also didn't represent the wide sort of geographic reach of our data set. The mixture of individuals, they were much smaller, much less heterogeneous. And so definitely that's something that our data set represents. It's something quite unique as far as its depth and breadth
Yeah.
of representation. Yep.
Now, I want to talk quickly about the Dream Challenge. We've talked about it already. It deals with this vaginal microbiome data set. Jonathan harmonized the 16S region, allowed you guys to harmonize the data that was taken from different regions of 16S.
Mm-hmm.
And scientists from around the world, experts from around the world, some of whom had nothing to do with preterm birth and didn't even know about preterm birth, got together in teams and made predictive models based on that data for preterm birth for various times of preterm birth. I think it was early and super
That's right.
early, right?
That's right.
So before
Exactly.
37, or I think before 34. And these models helped advance understanding of vaginal microbiome driven preterm birth, which is actually one of the leading causes of spontaneous early labor. Some experts within our network put it at something like 35 or 40%, Tomiko. So this is a huge one. And when I say that these models helped advance understanding of vaginal microbiome during preterm birth, Tomiko, what do I mean? What did we learn and gain as a field from these models? So wonderful. We've got the data. Your team cracked it. We put it out there to the community. They gave their models. They had a high degree of accuracy. But what did we learn about vaginal microbiome driven preterm birth from this work?
Absolutely. So anytime you do machine learning, one of the things you can look into, if you have good predictive performance, you can look into the features that contributed to the prediction of your model's performance. You can look into these important features. And that's what we did. So when we first tried to apply our own prediction models, let's just say our performance was nowhere near as good as what our challenge participants were able to achieve. And so there's not too much we could have learned from our baseline work, but our challenge participants were able to achieve much better performance in the prediction than we were. And then we were able to look into the important features of their models, both individually, and then we also looked at it like combined performance across the stronger models, and then looked at those important features. So we looked at it in different ways. And these insights that we gained by looking into the important features gave us some clues into which particular organisms may be more strongly associated with preterm birth, which ones are contributing to this signal that we're seeing in the predictions. So these are helping to validate things that have already been recognized in these simpler association studies. The fact that we're able to pick up on these in machine learning lets us know that there's something there for the predictions. And maybe, right, as you know, right now, there's no tool. There's nothing right now that exists that can reliably and accurately predict whether individuals are at risk for preterm birth. And this tells us there's something potentially here. And maybe this could translate into a novel, diagnostic or some kind of predictive tool. There's definitely something here. And that's what we were able to learn from this project.
Yeah, and the thing that I heard you say, Tomiko, that I just kind of hooked onto is that this was kind of a validation of previous work and previous associations that other
Hmm.
researchers, and certainly within our network, and absolutely at Imperial College London, because those guys are really leading the fight here against vaginal microbiome during preterm birth, have already made. And so, Tomiko, is it correct then for me to assume that some of these features that were interesting that you found basically showed that it was the Lactobacillus crispatus dominant microbiomes that had lower risk of preterm birth and maybe lactobacillus inners, or is it the Gardnerella, the one that leads to poorer outcomes? I
That's
forget
the other
what the
way.
strain of that one
Yeah,
is.
very good. Yeah, no, you've been following for sure. No,
I love that
absolutely.
topic.
Yeah, no, it's also near and dear to our hearts. Yeah, no, definitely.
Yes.
We saw that there were certain organisms belonging to certain community state types, and those are like groupings of these
Yeah.
bacteria by certain by their identities. And there are certain CSTs that are more associated more strongly with better outcomes, and others that are associated with what with worse outcomes. And as you noted, there are certain ones that we saw that were associated with better with not preterm birth, but more
Hmm.
like contributing to term birth, and
Hmm.
vice versa. We saw the sort of the classic bad guys among these bugs, these microbiome that were popping up, that were contributing to the prediction of adverse outcomes, such as early preterm birth and preterm birth. So it was definitely reassuring to see these novel and known organisms surfacing in this work.
Yeah, absolutely. Now let's move on to another piece of news from the database. So we're moving away from NAIR, and we're going to talk about something that's really exciting, which is the launch of ChatPTB. I did not say Chat GPT. I said Chat PTB, Chat Preterm Birth, the first preterm birth research AI assistant in the field. I should mention Chat PTB uses Chat GPT and other technologies to answer questions about preterm birth, but you would say it might be have a little bit of a sharper focus, right? It's different from Chat GPT. How, Tomiko?
Yeah, no, absolutely. So to give a bit of context into what this is, we decided that we have, as you mentioned, we have all of the prematurity research centers data and publications represented on our website. And for any individual to come and try to parse all of that information, it would take them quite a bit of time. I can't imagine how long it would take someone to read a few dozen articles, let alone look at across all of the study data. So as we know now, we have large language models that are able to really like analyze and provide information about these very large, texts. So we thought, well, why don't we leverage ChatGPT and apply this to our data website, and let individuals get answers on our publications and our data. The name, we have to credit Jonathan Cherry. He came up with this brilliant name. And ChatPTB does this. What it does is it searches across our papers and gives people answers to questions that they may have about our research work or anything related to preterm birth. And the nice thing is that we've been very thoughtful in the design of this to make sure that, as we know, Chat GPT can hallucinate. It can give you information that's not trustworthy, but we've made sure to focus on ensuring that the responses that people get are accurate, reliable.
Okay, so one of the reasons that the answers might be more accurate and more reliable than Chat GPT is because there's potentially less hallucination, and that happens as a result of too much data. Is that right?
That's well, exactly like when Chat GPT is not doing its work carefully, let's say, it will hallucinate and it'll fabricate responses. And that is not good for any of us. And so what we made sure to do was include something called retrieval augmented generation. It's an AI technique that that enhances large language models by retrieving relevant external data from known knowledge bases before it generates a response. So it basically allows the large language model to get fresh facts before it answers a question. And that's what really helped us ensure that we have better accuracy. It
And
also
those,
will cite sources. Yeah, exactly, to make sure that people can then check to see where the information is coming from.
okay, and the information is only coming from the database.
Yes, exactly.
So
Right
it's
now,
so it's kind of like a super filter that you put on ChatGPT
that's right. That's right.
because we trust
Exactly.
the
So
quality of our work.
exactly, we do. And we want to make sure that what people are getting is a good representation of that and not something that's filled with nonsensical information.
Yeah, absolutely. AI hallucinations sounds pretty spooky. When you first said that to me, I thought you were making a joke, and I thought it was really good. And it kind of is a joke, but it's realistic. And we've all been there. We've all used ChatGPT and said, "How did you come up with that?"
Yeah, definitely. Yeah, no, I agree with you. Hallucinations, it's kind of funny. I want to say it's more like confabulation, where it's like making stuff up versus like
Hmm.
seeing things that aren't there. But exactly, AI hallucinations are like made up facts, or with fake citations or incorrect calculations. It's invented detail. But the scary thing is, it sounds believable. It sounds like it could be real, and it's not until you dig in that you realize, oh my goodness, this is actually absolutely wrong. So that's
Right,
sometimes
right.
you're right. So it's scary in that sense, is that it sounds believable, but it's
So
not
you've still got to dig in. You've still got to dig in.
always, always.
Now, Tomiko, does there I've seen the papers that go into the database. They've got charts, they've got graphs, they've got numbers, and they've got text. How much of that is included in ChatGPT's answers? I'm sorry, ChatPTB, does the assistant read those graphs accurately and give you insights based on visual scientific presentations, or is it just text?
You're getting digging into the details. You're absolutely right. It is currently just pulling text data. So it's
Okay.
not necessarily looking at the images or other, I don't even think it's looking at attachments. It is pulling data from
Mm,
PubMed, and that information is text. And so right now, this is trained to provide answers based on the text that it has from PubMed regarding our articles. The hope is, though, this is not like everything that we have on the website. It undergoes refinement, iteration, improvement. So the goal is to expand on this and to include additional sources of data. And that would make this tool even richer than it is now.
yeah, absolutely. Do you yourself use the tool?
I do. I do.
Really?
It's funny. Like, yeah, no, I do, because I too, like anyone, at first, if there's a publication or two or three or 10, you remember. When you start hitting like 80, 100, it's hard to remember where certain pieces of information are coming from. So I do a quick check just to make sure, I mean, why not? We've got these tools available to us nowadays. So absolutely, I am using it.
Can you give us a couple of examples, one or two, of the kinds of questions you're asking ChatPTB?
Yeah, it's more like currently I've used it more for just confirmation of information and where it is that I've gotten it from. So just asking about where was XYZ fact, um, you know, published again, and it will tell me, and the answers have been correct. Um, I have not used it. I guess I know which research questions we're exploring. I haven't used it beyond that, but I know our colleagues across the prematurity research centers are currently helping us stress test this and asking their novel questions. And so I look forward to hearing their feedback.
And so you can ask it questions like, what are the causes of preterm birth, right? And it might not
Sure.
give you, it might say, we're not sure yet, but here's what we know are associations, right? Because we know that if,
Exactly.
if we had an answer to that question, we may not have this podcast. But I guess what I'm trying to say is that you can ask very simple, large questions about preterm birth, what are risk factors, et cetera, to really nitty gritty questions about some of the about the papers that we have published as part of the PRC.
That's right. And people they've tested questions that have asked more broadly, not specifically like where does this fact come from, but more broadly like um, what are some features associated with preterm birth? What are some organisms that are associated? And that kind of information comes across multiple publications. And we've seen that the responses that it gives were partially checking to see what is this information, and also partially fact checking to make sure that there aren't hallucinations that are affecting this tool. And we've been pleasantly surprised that this is working well.
Tomiko, why launch this tool? Why ChatPTB at all?
I feel, why not leverage large language models to lower the barrier, lower the entry level for people to come in and help us with discovering novel answers for preterm birth questions. I think there are people who want to help. As we found with the AI challenge, when we found with the Dream Challenge, there are people who want to be involved, but they may not know, or they're brought in by opportunities like this that come about. So our hope is that by offering a tool like this, we can have people who are interested ask questions of our data. Like, what are some ideas that they can come across? What are some ideas that they might be thinking about that might be answered with our data? And on the same page as our ChatPTB tool is also a set of tutorials for how to use AI, how to leverage ChatGPT. Because as we know, ChatGPT can do more than give answers. It can actually help you code. So if you have a question and you come to this website and you ask ChatPTB, what's something, something that's interested of me, like, are there drugs that might be able to help with, treating preterm birth? And it gives answers based on our data. And then people say, okay, well, I have an idea and I want to do something, but I don't know how to code. Well, they can use our tutorials to then follow up on asking their questions. They can even use our tutorials to work with our data. So I guess we're trying to provide a gateway that's easy for people to use to come and join us in this work against trying to find ways of helping people who are suffering from preterm birth, and help us find solutions.
Yeah, you're making it really easy. You're rolling out the red carpet. So what's next for the database, Tomiko, for the AI assistant and for you and Marina? What is on the horizon? Because you've said multiple times during the interview that time goes so fast, and in the blink of an eye will be another year or 2 or 3 years into the future. What does that future look like?
Yeah, no. So if I blink now and I open my eyes in a year, I'm hoping that by then we'll have the inclusion of additional types of data. As you mentioned, right now this is text-based, but the hope is to expand it beyond that. And that's for ChatPTB. And also, we know that co-scientists are something that are now helping people. These are, AI methods that are specifically more for science. And so what we have now on our website is using ChatGPT, which is more of a general tool. But if we can use these co-scientists to really, if we can not just tell people about their availability, but kind of provide tutorials on how to leverage these resources, then again, our hope is that, we're all in this work, but we want to help bring others into this work. So just expanding on the website to make more information available, and the hope is that and one easy way that we can kind of see this is the number of downloads of our data sets. My hope is that if I, again, blink and open my eyes in a year, that we're going to see this rapid rise in the amount of downloads from our website. Because again, we want people to be using our data. We are all for open science and bringing everybody in to work with our tools and our resources to help us in this fight against preterm birth.
Well, Tomiko, you have been lovely. Thank you for coming on the show, and
As have you.
congratulations
Thank you.
again.
Thank
Congratulations
you.
again, both to you and Marina, on this year's wins, which are much deserved. I know that both of you are incredibly hardworking women scientists, and you should be proud of yourselves. And you both put your heart and soul into this database every single day, and the PRC, all in the name of mothers and babies and ending this crisis. So thank you.
Well, thank you for featuring our work. We really appreciate it.
Thanks, Tomiko.